I'm Mr. Davenport, and this is uh, our Introduction to Biotechnology course. Today, we are taking a look at the historical applications of biotechnology. So, biotechnology is honestly a really old technology. Just humans didn't have the term for it when it was originally applied. Humans have been using living tools from plants to microorganisms for thousands of years. And today we're going to take a look at some of the notable uses of biotechnology throughout history. So, guys, you might want to have a pen and paper out to write down the dates and the events that happened in these historical applications of biotechnology. Today, ladies and gentlemen, we are going to be utilizing a timeline called TikiTaki.com. I am going to be adding this into Canvas for you to be able to use some of the external tools and videos and images that I might not be exactly touching on in this presentation. Today, I just want to go over some of the big impacts and applications of biotechnology through the, well, several thousand years. So we're starting way back in 750 BC when ancient peoples, including Sumerians, were the first to brew beer. Ancient peoples used a microorganism called yeast. Yeast basically survives by eating, well, sugar. And so when you add yeast to a starchy mixture of wheat or corn or whatever, it begins to ferment. And the outcome of fermentation is alcohol. What this alcohol did is it preserved the liquid mixture, the malt, and slowly turned it into a drinkable liquid called beer. Now, the reason that this is important, beer and wine was important in ancient times, is people really didn't take great care of their water sources. They knew that running water was okay to drink, but they would also sometimes drink through puddles and still water, and this stuff could get easily contaminated with microorganisms. And because they didn't understand that the microorganisms could infect their body with dangerous diseases that could lead to illness, essentially what happened is ancient people relied on drinks like alcohol and wine as a way to have an alternative water source so that when they traveled, they would be able to know that they had something that would not make them sick when they drank it. All right, so moving forward to the ancient Greeks. So, Hippocrates. So, Hippocrates was considered the first doctor. Now, he really wasn't the first doctor. He was just the guy who created the first school of medicine. And he developed these standards of ethics and wrote the Hippocratic Oath, which is basically the oath of doctors to do no harm. And so Hippocrates was born in Greece in 460 BC, and his doctor was a physician. And as a young man, Hippocrates traveled the world, or what was known of the world at the time, and learned about medicine. Hippocrates studied the human body and made his observations of how it worked. And he developed his own theories of how illness, well, hurt people. And he rejected the current belief that was prevalent at the time in ancient Greece and all over the world, really, that illness was caused by evil spirits or by making the gods angry. And so, with these theories that he had, Hippocrates eventually founded his own medical school on the island of Kos and began teaching his ideas. And he taught that the body should be treated as a whole living thing. And he believed that treating illness with rest, a good diet, fresh air, and cleanliness was vital. And so Hippocrates is known as the father of medicine and credited with developing the ideas and principles known as the Hippocratic Oath that's taken by nurses and physicians today before they begin medical practice. So the vital part about Hippocrates is he was one of the first individuals to realize that medicine was an illness was not caused by some kind of punishment or evil spirit. That illness was caused by something that impacted people from the outside. People got sick because they were exposed to something different. They were exposed to something that was making them sick. Now, we're going to take a big jump and go into 1797. Okay? So, in 1797, Dr. Edward Jenner inoculated a child with a viral vaccine to protect them from smallpox. What happened is Edward Jenner discovered that cowpox, a less deadly disease that affected dairymaids, had some similar symptoms to smallpox, but not as severe. And that people who got cowpox didn't get this really devastating disease called smallpox. So he created the first vaccine by giving patients cowpox before they got smallpox. Now, so I want to basically take a moment and actually show you guys a quick video that I think is vital to understanding Edward Jenner's work.
Ten 10,000 years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her odd treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected, but it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairymaids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family, but when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the dairymaid Sarah Nelms, Blossom the cow, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
So, now, guys, that is the story of Edward Jenner. And up next, we are bringing in one of his contemporaries. Not long after that, we had Gregor Mendel. Gregor Mendel basically discovered genetics, and he did this by studying pea plants. He discovered that the traits of pea plants were passed from parent plants to seeds to plants that were planted from the seeds. And these were discrete or hidden independent units that we later identified as genes. It was Gregor Mendel's research that laid the groundwork for the field of genetics, which is the study of DNA as it relates to heredity. Now, Gregor Mendel was born in 1822 to Austrian poor parents, and at the age of 21, he joined the monastery, and he wanted to study philosophy and theology and natural science. While at the monastery, Mendel experimented with flowers in the monastery's garden, trying to develop different color variations. And it was in these experiments that led him to what we would call hybridization. That means creating a hybrid, a combination of two or more things. And so between 1856 and 1863, he cultivated and tested almost 28,000 plants. He did much of this research with pea plants because they were easy to obtain and to grow. Mendel's work wasn't really understood during his lifetime, and it wasn't until the 1900s that 3 scientists from different countries in Europe did similar experiments and came across the research that Gregor Mendel had done. Through his remarkable research in plant hybrids, Mendel established that there does exist living things dominant and recessive characteristics, which is why we call what we call the Mendelian law. He is often called the father of genetics, and ultimately he passed away in Austria in 1884. But the main thing that out of that story is that Gregor Mendel was responsible for discovering what we now call genetics. Now, next is Louis Pasteur. Louis Pasteur develops the first rabies vaccine. And so Pasteur was born in a small town in France in 1822. His father was a leather tanner. That means he took the hides off of animals and turned them into leather, which was used for a variety of reasons in the 1800s as it is used now. Now, Pasteur was a pretty smart kid, and by 1847 he had already earned his doctorate in physics and chemistry. And he continued his research for many years and is credited with a number of different discoveries. Pasteur founded the science of what we call microbiology, the study of microscopic living things. And he proved the germ theory of disease that Hippocrates had in his mind back in ancient Greece. And in the process of proving germ theory, he developed this process called pasteurization. Pasteurization is heating up a food item to the point where it would be a boiling point, almost just below it, to kill millions of bacteria to help prevent it from getting spoiled. So when it says on your milk and your cheese pasteurized, it means it's gone through this process to kill off this bacteria that comes naturally within those foods. So we kill the bacteria so the milk and the cheese will last longer. He also discovered that weakened forms of a microorganism can be given to a person who will then develop their own immunity to a disease. Basically, he developed vaccines. Edward Jenner was the first one to test out vaccination, and Louis Pasteur brought it into the modern era. As a result, he developed vaccines for several diseases, including rabies, and Pasteur is responsible for influencing changes in hospital practices to minimize the spread of microbes. But the main thing that we want to take away here is that Pasteur developed the first rabies vaccine in 1883. And then we go into 1906, the term genetics is introduced. The word gene was derived from the Greek word genesis, meaning the truth, or genos, meaning origin. Now, Alexander Fleming discovers the penicillin, the first antibiotic. So here's the thing about 1928. It's also the year that FFA was founded. But this is the year that Alexander Fleming discovered penicillin. Now, penicillin is a mold, okay? It's a fungus. And the thing of it is that this penicillin bacteria kills penicillin mold, fungus, kills other microorganisms that try to take up its space. It creates toxins. And so these toxins are dangerous to microorganisms, but not dangerous to macroorganisms, not dangerous to humans and animals unless they have an allergy. And so Alexander Fleming, through a pure accident, because his lab was kind of disorganized and not really tidy, he decided to leave some dishes where he was growing bacteria, staphylococci, so staph infection, in the wash and the bleach solution, basically. Well, one of those plates didn't get cleaned very thoroughly, and a bread mold fungus fell onto one of the open plates where the bacteria was growing and sprouted and bloomed and grew. And as it grew, well, Alexander Fleming had gone on vacation. And when he got back, he realized that there was this big gap between where the mold was growing and where the staphylococci bacteria was growing. almost like something had killed the bacteria and poisoned it. So he tested it and found out that this Staphylococci bacteria was literally broken apart, torn apart by this mold. So he set about to test his theory, grew a whole bunch of this bread mold, Penicillium notatum, and grew this and tested it, mold juice, okay, called penicillin, and tested it against bacteria. And eventually he turned it into a shot that would be injected to treat disease that was caused by bacterial infections. And penicillin is still used today as a treatment for bacterial infections. All the way back in 1928, Alexander Fleming discovered penicillin, the first antibiotic. So, guys, this has gone for a little over 20 minutes. I'm going to stop right here, and I'm going to pick it back up in a little bit later and hopefully get through the rest of this lesson. See you in a bit.